Okay, so just as a review over what we talked about last week, remember that we are in chapter 17 of your book and we're talking about blood. So where are blood cells produced? Actually, that's not a right question, is it? Blood cells are produced. And why do I have that number? Blood cells. How about where are they broken down? Where are the blood cells broken down? Does anybody know? Not in the liver. The liver stores iron. Not in the kidney. The kidney produces a hormone called EPO, which stimulates red blood cell production. Should be in the bone marrow, right? So in the bones. Oh, would be bones. Sorry, I was looking for bone marrow. Okay. So the spleen is where the red blood cells are actually broken down. Okay. They're produced in the bones. Okay. So is cholesterol water soluble or water insoluble? Insoluble, right? Because it's a lipid. But we have to transport it in the blood. So how is it transported in the blood if it's not soluble in water? What must be in the blood in order to transport it? It's not an enzyme because it doesn't break it down, but it transports it. Does anybody remember what we talked about? So we talked about lipoproteins. And so lipoproteins are in the blood and they transport cholesterol. And so when we talk about high density versus low density or HDL and LDL lipoproteins, that's what we're referring to. So there's really only one type of cholesterol. So there's no good or bad cholesterol, but it has to do with the lipoproteins that transport the cholesterol. And interestingly enough, estrogen and testosterone are also lipids, and so they do not go into solution either. So you actually have proteins in your blood that ferry estrogen around and actually help to transport testosterone as well. And so anything that is a lipid cannot be transported directly in the blood. It has to be generally bound to a protein. So hemoglobin is found in which of these? The erythrocytes, right? So the blood plasma is everything but the cells. So the erythrocytes are the red blood cells. So hemoglobin is our respiratory pigment. It is the reason why our blood is red. And it is specifically packaged into erythrocytes. So do erythrocytes have nuclei or no nuclei? No nucleus, right? And so they actually start out being nucleated. And then as they go through the process of specialization, they actually eject their nucleus so that they can get more hemoglobin and store it in the cell, okay? So we're going to talk about that, the life cycle of the erythrocytes in a minute, okay? So which athletes would tend to have a higher red blood cell count? One that is training in Los Angeles or Denver? Denver. And what does that have to do with? Altitude, but what is it at the higher altitude? What has to do with oxygen? Is oxygen at a greater concentration, up high or low? Low. Low, right? So when you're up high, right, the oxygen concentration in the air is lower. And so in order to get that transported, they would actually increase the red blood cell count, right? So that they would actually be able to carry more oxygen and pick up more oxygen in the lungs. Okay, so that has to do with that EPO. Okay, so today we're going to talk about red blood cells a little bit more, and then we're going to start talking about white blood cells, and then we're going to finish the chapter on blood, talking about transfusions and blood typing. So when we look at the life cycle of a red blood cell, because it doesn't have a nucleus, it doesn't tend to live very long. And so it has a life cycle, let's get the right pen here, of about 100 to 120 days. So what happens is that the red blood cell will start to get damaged, and it'll start to kind of break down and it'll kind of change shape. And when it changes shape, what happens is it starts to get clogged into the capillaries. And specifically, it gets clogged in the spleen. because the blood is filtered through the spleen. And you'll remember that in the spleen, we have a special type of connective tissue called reticular connective tissue, which means that there's lots of fibers, right, in that connective tissue. So they get broken down by macrophages in the spleen. So macrophages, especially, I'll just put especially in the spleen, because they get kind of stuck in there. There are also macrophages in our blood that can break down damaged red blood cells as well. So we have to store iron, so we recycle the components. So we store iron in the liver. And we can't store iron. Remember that iron is actually toxic. And so we have to store it as bound to other atoms. So it is either as ferritin or hemoceridian. Hemoceridian. Siderin, sorry, siderin. Siderin, there we go. Okay. That's an N on the end of hemosiderin. Okay. Then remember that the hemoglobin, when it is broken down, it produces bilirubin. So we talked about newborn babies and having to break down fetal hemoglobin, and how that sometimes causes bilirubin levels in the skin to become elevated, and they get what is referred to as jaundice. So bilirubin is actually transported and leaves the body with the feces. So it has a component of bile, so it is part of bile. And it gives the feces its color. So if you don't have bile, if you're not producing adequate levels of bile, then your feces will tend to be really light in color, could be even like maybe tan or whitish in color. Okay, so the more bilirubin you're breaking down and transporting into the small intestine in the form of bile, the darker the feces will be. Okay. Also, sometimes what you eat can affect the color of the feces too, like beets, for example. Okay, so that's bilirubin. And then we have, remember, we have the proteins. And so remember that in adult hemoglobin, it is alpha and beta globulins, or globins, actually, globins. Okay. And so these are broken down into amino acids, and the amino acids are passed back into the circulatory system. So they go back into the circulatory system where they can be actually transported, you know, maybe back to the bone marrow or out to the muscles. So those amino acids are necessary for protein production, and so they can be recycled from the hemoglobin that is broken down. Okay. So if we look at a diagram that shows this whole process, and this might be a diagram that could appear on a quiz. Remember, your quiz is on Wednesday. It would look something like this, right? And so here they're just showing the active role of the kidney in causing the production of red blood cells, which we talked about last time, which is called erythropoiesis, and how these erythrocytes are then released into circulation. And then this is breaking it down. So here the bilirubin, right, is secreted into the intestine, right, as bile. Here the iron can be stored in the liver, or it can be drawn out of the liver and passed into the blood. And then the globins are broken down as amino acids and passed back into the circulatory system. And so this just shows the life cycle, the birth and the death of the components of the red blood cell. Okay, so the next thing we're going to talk about are the leukocytes. And so remember that the leuko actually means white. So these are the white blood cells, right? Not the red blood cells. And also remember that when we centrifuge whole blood, that the red blood cells go to the bottom, right? And then the white blood cells are in the middle part, and that's called the buffy coat. And so this makes up only about 1% of whole blood. What is the average percent that the red blood cells make up? Does anybody remember? Is it like 20%? Is it like 30%? Is it like 80%? 45, yeah. So remember that the hematocrit value is the percentage of whole blood that is red blood cells. And so this is not the hematocrit value, but this is the size of the buffy coat. So this is the buffy coat. And we're going to do a blood lab on Friday, where we're going to look at our hematocrit values. And we're also going to test our blood for blood typing. So when we look at the leukocytes, this is also produced by the bone marrow, but the stimulation of the production of leukocytes can take place in the blood and in the tissues. So I'm going to talk about the types of leukocytes, and then we're going to talk about their production. So when we look at the leukocytes, there is an easy way to remember the order of abundance. So we'll put order of abundance. And there's a mnemonic device where you can just remember, never let monkeys eat bananas. Okay, so these first letters of these words are the 5 different types of leukocytes that we have in our body. So what do you think the N stands for? Does anybody know? From previous maybe biology classes. This is the most abundant white blood cell in your body, and they are called neutrophils. Okay, so most abundant leukocytes. But remember that the red blood cells outnumber the white blood cells by huge numbers, right? So it's actually called a neutrophil because of the way it picks up stain, because it picks up both types of stains, so pink and blue. And so neutrophils tend to look purple when you look at them on the slide of the blood, because they pick up acidic and basic stains. So you might hear about these when you take microbiology. So when we look at the neutrophils, these are the types of blood cells that are called phagocytes. So phago means eat, right? So these eat bacteria and other debris. So if there's like broken down cells or other things, they would take those in. So phage means to eat. So the neutrophils are the phagocytes. And specifically, these are the types of white blood cells that would respond immediately to injury, to like infection. So if you get scratched or if you get an infection, the neutrophils will be right on it. So they respond and increase their numbers in response to infection. Okay. Now I'm going to skip over the L and the M because there's another way that these cells are classified, and that is if they have granules inside of them. And so I'm just going to put granules present. And what this means is that there are lysozymes with enzymes. So a lysozyme, actually lysosomes, we'll put lysosomes. Lysosomes and enzymes. So the lysosome is a membrane-bound structure in your cell that you might have learned out about if you took Biology 112, right? And there's enzymes in there that help to break down. So once the neutrophil has taken in the bacteria, then it will start to digest it, okay? So I'm just going to show you a short little video that shows the movement of the neutrophils. Neutrophils can leave the circulatory system and go out into the tissues, and they are attracted to chemicals, and so they can actually kind of track and move towards bacteria. So oops, that didn't work.
Neutrophils are white blood cells that hunt and kill bacteria. In this thread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have bananas. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil.
Maybe if I make it smaller.
The neutrophil becomes polarized and starts chasing the bacteria. The bacteria bounce.
So these would be red blood cells. This is the size of a bacteria in response in order to distinguish between white blood cells.
And engulfs them by phagocytosis.
Okay, so it takes the bacteria in, and then the food vacuole fuses with the lysosome, the enzymes are released, and then the white blood cell can actually feed on and eat the bacteria. Okay, so if you have an infection, you're more likely to have an elevated count of neutrophils than any other types of white blood cells. Any questions about that idea? No. Okay. So you notice that it is moving like an amoeba, right? And it also is moving in response to a chemical trail. So it tends to move in response to chemicals that are produced by the bacteria. Okay. So the next group of white blood cells that I want to talk about are the eosinophils. Okay. These are also granulocytes, but instead of fighting off bacteria, these are actually cells that are responsible for detecting and fighting off multicellular pathogens or even multicellular parasites. Okay, so this would be respond and attack multicellular parasites. Okay. So some examples of multicellular parasites might be, right, hookworms, hookworms were actually quite prevalent in the United States in the early part of the 19th century. The most prevalent parasite that we have right now is called a pinworm. And pinworms are common in children. And then obviously a tapeworm would be another example of a multicellular parasite. Okay. So the other thing that the eosinophils are responsible for is attacking allergens. So these respond to allergens. So an allergen is anything that causes an allergic response by the body. And allergens are not something that is actually going to cause damage to us. So whereas these parasites could cause us damage, like hookworms could give us anemia because the adults live in our intestine and feed off our blood. And tapeworms also can make us really skinny because they absorb the nutrients across the surface of their body and they reside in our intestines. The allergens are not dangerous, so substances that pose us no harm. Okay, so an allergen tends to be something that would not normally be something that would actually invade and infect our body. And so an example of this would be pollen from plants, right? Cat dander or animal dander. The worst one to have is, which actually I have this one, which is dust mite feces. Dust mites are everywhere, right? So they say, when I went to the doctor, he says, oh no, I'm sorry. This is year-round allergies, right? It's not just during, he also said, oh, you're allergic to wheat pollen. Oh, that's really nice. And I'm really allergic to horses, for example, and specifically horse dander. Okay, so one of the interesting things about this is that this can give us allergies and even lead to asthma. And asthma is a constriction of the bronchioles, so that when you breathe it in, it's kind of irritated and your bronchioles constrict and you can't get enough air, right, into the respiratory system. And so there is a hypothesis here that we have actually eliminated some of the parasites that our immune system co-evolved with. So we evolved having parasites in our body, and just like the bacteria, perhaps those parasites now, if we completely remove them, are responsible for our immune system being hypersensitive to other things like allergens, right? And so the hypothesis is called the hygiene hypothesis. Is that we are too clean and we lack parasites. So our immune system responds inappropriately. Another thing that can come about by our immune system attacking our own body, and that would be autoimmune diseases. So we already talked about one of these related to joints. Does anybody remember what that was? Rheumatoid arthritis, right? So this could be the fact due to that our immune system has nothing to fight. And so the eosinophils are responding to healthy, normal tissue inappropriately and attacking it, right? Because we've eliminated our parasites. And so some people, and I ran out of room here, but I'll write it up here. Some people are using what is called hookworm therapy. And what this is deliberately infecting yourself with hookworm larvae that then get into your digestive tract, and then your immune system has something to fight. And what they're finding is with this hookworm therapy is that it decreases allergies, it decreases asthma, and in some cases it's actually decreasing the symptoms of the autoimmune diseases. And some of the autoimmune diseases are horrible, like Crohn's, where it's your digestive tract which is being attacked, and then lupus, where it's like all over your body. And so the hookworm therapy, the idea is that once you get, like, say, 10 adult hookworms in your body, they can't reproduce inside of your body. They actually have to come out and then reinfect you. And because we have a sanitary system that would kill the eggs when they come out of us. We don't, you know, have feces on the soil. So the idea is that once you get 10 hookworms, then you're not going to get any more. And then they're going to live like maybe 3 to 5 years inside of you, and then they die. And then you would, if it wanted to, you could get more hookworms. And so the idea is that you can control the infection, and you can control the number of hookworms you have in your body, yes.
Does it sound under system response?
The system responds inappropriately.
Will
Yeah.
there be some really big side effects to the hormone therapy? Otherwise, like,
What?
there has to be like some really serious side effects for the hormone therapy. Otherwise, like, everything would do that.
You know, people are freaked out by thinking that they could have parasites inside of them. So I don't think that everybody would purposefully infect themselves with a parasite. Maybe if I had, because I have really bad allergies, I can imagine what my life would be like without having to take allergy medicine every day of my life. I'd be like, that would be really nice, right? The bad side effects was when we were not controlling the infection rate. So, like, for example, before we had bathrooms, indoor bathrooms, we had outhouses, right? And sometimes the feces could get into the soil. And so a person can have like 100 hookworms in them. And the side effects is anemia, because those hookworms are in your gut and they're feeding on your blood. And so your side effects would be that you would be anemic, right? And you'd feel really bad.
But nowadays, this would be great.
Some people, there's research, there's internet stuff on it where the people have infected themselves. Sometimes they say they can feel pain in their intestine. So they think that they can feel them latching on, and that could be a potential source of pain. But not having too many, they would not be anemic.
What a doctor would do that.
Not in the United States, right? In Europe, I think in here you have to go to Mexico. So you can go down to Mexico and get your helmets. Yeah. Question?
No, you answered it.
Okay. Okay, so those are the eosinophils, and these are also what are referred to as granulocytes. Right? So granulocytes, they have granules. Okay, granulocytes. Okay, so never let monkeys eat bananas. Let's go to the last one, which are referred to as basophils. Okay, so I'll just put this down here. Okay. So basophils are actually the least abundant white blood cells because they're the end of that mnemonic device, bananas, right? And you can see that the basophils also have granules. So the basophils have granules filled with histamine. And histamine is a chemical that causes capillaries to become leaky. So histamine causes capillaries to become leaky. And so fluid, including white blood cells, get into the tissues. And so this is what causes tissue swelling. Right? So when your nose gets all stuffed up or your eyes start to itch, that has to do with histamine, right? And thankfully, we have antihistamines to fight them, right? So over-the-counter antihistamines like Benadryl, right? This blocks the action of histamine. Okay, so they always recommend that you have Benadryl, which is a little bit faster acting than, say, for example, the Allegra, or what are some of the other ones? I can't think of the names, but anyway, the Benadryl on hand, just in case you have a allergic reaction to something and you have to take it quickly. Because one of the problems with this is it could actually swell up your throat and your pharynx and your nasal passages so much that you wouldn't be able to breathe, right? So Benadryl can help with that. So this would also be a granulocyte. And then let's look at the other ones. So never let, those would be lymphocytes. Okay, so L stands with lymphocyte. And these last two ones would be agranulocytes. So I'll put a little parentheses here. And I'm going to put monocytes down here. And these are called agranulocytes. Because remember that granulocytes. Remember that if you put, that's not T, granulocytes there. Remember if you put an A in front of something, it means that it's without, right? So like asexual, without sex, right? Agranulo, and this is one word, site, that would be the lymphocytes and the monocytes. So you might know the lymphocytes if you studied the immune system previously, because this includes T cells. And T cells are most famous for the fact that the HIV virus that leads to AIDS attacks T cells, a specific type of T cell called the helper T cell, and that causes your immune system to like crash, okay? And then we also have B cells. So these two types of cells have distinct functions. The T cells attack and kill viral infected cells. So virus infected cells. Right? They also attack and kill cancerous cells. So they actually release enzymes that punch holes in cells, and then they release the enzymes that then go in and digest the cell from the inside out. So it's kind of like bullets. They actually create holes in the cells that they are attacking. Okay, so B cells are very different. So B cells produce plasma cells in response to infection. And plasma cells produce antibodies. Okay, so remember we talked about antibodies just a little bit. We're going to talk about more when we start talking about different blood types, right? But the antibodies are not antibiotics, right? They're Y-shaped molecules that have specific binding sites that can bind to a specific pathogen. So sometimes they call this the T cell and the B cell. This is acquired immunity. This is how you learn over your lifespan to detect particular pathogens and then fight them off quickly. Okay. It's also for the whole basis for vaccination, right? You can present your immune system, the T cells and the B cells, with a particular type of pathogen, and then your immune system will learn how to produce antibodies in response to that, and will fight off the infection if you get, if you experience the actual microbe that causes the disease, like the actual flu virus, versus the weakened flu virus that they give you in the vaccination. Okay, so monocytes, these turn into macrophages. And these also feed on bacteria and damaged cells. So for example, our lymph nodes and our spleen are full of macrophages. And the macrophages, when they detect a pathogen, then they start to reproduce themselves, and so that's why your lymph nodes will swell when you have an infection, because of the presence and the reproduction of these macrophages. Okay, so if we look at the relative number, remember our mnemonic device is never let monkeys eat bananas. And so never, most abundant, these are the neutrophils. Let, lymphocytes, monkeys, monocytes, eat, eosinophils, and then the basophils are the least abundant type of white blood cell. Okay. We're going to look at the different types of cells in a lab this week as well. Okay, so leukopoiesis. This is similar to erythropoiesis, right? Which has to do with the production of blood cells. So this is specifically the production of white blood cells, right? And so these are derived from stem cells. In the bone marrow. So remember that erythropoiesis is controlled by erythropoietin. Leukopoiesis is a little bit different. This can also actually be controlled by other white blood cells. So we can say that other white blood cells, or other leukocytes, I'll put other leukocytes, can stimulate leukocyte production by producing chemicals called cytokines. So these are chemicals that are released if a cell detects a pathogen or a virus. So let's say we have a T cell and it detects a virus in your body. It will release a cytokine, and then that would cause more of those specific types of T cells to be produced. And so we actually have two different types of cytokines, two categories. And these are called interleukins. One category is. So just look at what this word means. Leukin would be white blood cell, and inter would be communication between white blood cells. Okay, so interleukins can cause white blood cell proliferation, as can colony stimulating factors. What interleukin means? So interleukins can communicate between, or chemicals that communicate between white blood cells and cause more white blood cells to be produced or to proliferate. So other leukocytes can stimulate leukophore producing cytokines. So there's two categories of cytokines, interleukins and colony stimulating factors. So in your book, they write those as CSFs, or colony stimulating factors. So sometimes you'll hear about treatments for disease that sometimes they try to treat diseases by using interleukins or CSFs. So sometimes those are, they're looking into them to see if we can like stimulate white blood cells to fight off and attack disease if we give a person that particular interleukin or colony stimulating factor. So if you've had like the flu, can you give yourself interleukins to cause more white blood cells to be produced so that you can fight off the infection faster and not get so sick? Okay. So in your book, they have a picture. You do not need to know the derivative of it, but I just wanted to show you, right, that there's actually two different types of lineages of white blood cells. One is the lymphoid stem cell, and you notice that they have an interesting, just a spherical nuclei here, and then the myeloid stem cells that give rise to the granulocytes. Oops, these are the granulocytes. And then these would be the agranulocytes. And then some of the agranulocytes, for example, become plasma cells, and those plasma cells would release antibodies. So this just shows kind of a flow chart of the production and specialization of the white blood cells from the stem cells that reside in the bone marrow. Okay, so let's look at some leukocyte disorders. So leukemia. Does anybody know what leukemia is? What is that? Cancer, right? So this is cancer of the blood cells, specifically leukocytes. So leukocytes reproduce uncontrollably. So what this generally ends in is that the bone marrow becomes packed with white blood cells. And then we also have immature leukocytes being found in the blood. It's kind of interesting because one type of leukemia tends to be more common in children. Sometimes they call that acute leukemia. So children typically get leukemia. And then some types of leukemia are actually more common in elderly. So when we look at a blood smear, right? Normal blood, you would see very few, right? Leukemia, you would see a lot of, and in this case, it's mainly there's lots of monocytes and neutrophils here, okay? So you have excessive numbers of white blood cells. You would also find, however, an elevated white blood cell count if you had an infection, right? So sometimes they go in there and they take a little sample of your blood, and then they look at it and they say, oh, well, this person has an infection because they have an elevated white blood cell count, right? And so then they would give you drugs, maybe antibiotics to help treat that infection. But leukemia, these cells have become cancerous and are just reproducing for the benefit of themselves, and so they don't generally perform their normal function. Okay. We could also have low leukocyte, so we could have leukopenia, which is low leukocyte count. So I mentioned a disease that would cause you to have low count. You remember what I said before? Today in today's lecture. There was a virus that specifically attacks white blood cells. Specifically, yes. So the HIV virus can lead to what is called AIDS. And AIDS is short for acquired immunodeficiency syndrome. Is there a C in there? I think there's immunodeficiency syndrome. So you'd have abnormally low numbers of white blood cells. There is another disease that we sometimes see, or illness, which is called mono, right? Which is sometimes referred to as the kissing disease, because it sometimes happens where you have this outbreak in teenagers, like in high schools, right? And this is infectious mononucleosis. This is not an example of leukopenia. This is another example of a leukocyte disorder. And they actually misnamed this. So it's actually not monocytes that become really abundant. It's actually lymphocytes that become really abundant. So they kind of misnamed it, but the name kind of stuck. So it's not lymphonucleosis, it's mononucleosis. So we have increase in lymphocytes. And this is a viral, due to a viral infection. So the problem is you can't take antibiotics for it because antibiotics are for bacteria. Oops, infection, viral infection. So it's one of those things that you just kind of have to wait out, right? And it makes people really sick. You know, they can have mono for like a month, right? And they're really tired and they're really weak and their whole body just aches. And that is due to a virus that infects and causes an increase in the lymphocytes. So I think I have a picture of that too. Let's see. Yes, so this would be, for example, they look like monocytes, but they're actually lymphocytes, so they can tell whether or not you have mononucleosis by looking at your blood. And if your blood looks like that, then they know you have mono. These would be neutrophils, so this could also be either a cancer or just that you are very, very sick. Okay, so the last thing we're going to talk about in terms of the blood is blood clotting. And so we're going to talk about platelets. And so platelets are cell fragments. And they are fragments of megakaryocytes. Okay. So megakaryo means big nucleus. And so these cells have a big nucleus and the cells just fragment off of them. And these platelets are important in blood coagulation. So coagulation means clotting. And that is going to be a little bit different than when we start talking about transfusion, mismatched transfusions, where it is actually called agglutination, not coagulation. Okay, so the platelets are involved in coagulation. So they can actually bind to damaged capillaries, so bind to the lining of damaged capillaries and stop blood flow. Okay, so let's say a capillary has been damaged and there's blood flowing out into the tissues. So you have hemorrhaging. So the capillaries actually have little receptors on them that will bind to the damaged lining of the capillaries, and then they'll just kind of form a clot. But the really important thing is that they initiate clotting of, we'll just say, initiate clotting cascade. Okay. So the clotting cascade is where we have all of these proteins that are in solution in our blood. And once this cascade is started, it causes the conversion of one molecule to a different molecule, and then that response causes another chemical reaction to occur. So that's why it's referred to as a cascade. And I'm going to actually show you a video about this cascade in a minute. Okay, but ultimately, this causes fibrinogen, which is a clotting factor. This is, I think I mentioned this before, this is soluble in water. And then this gets converted to fibrin. and this is insoluble. So these are the fibers that help to bind the platelets and also prevent blood loss. So it's actually the fibrin, the platelets, and then they're working together to form a blood clot. Okay, so let's see if I have a picture of that. Oh. So this is the pathway for the production of the platelets. You just need to know about the megakaryocyte, right? It's got a huge nucleus, and then it just blebs off these cell fragments. And let's see if I can find there's my clotting cascade. Okay, so this is what I wanted to show. So this is showing a blood clot where you have these fibrin molecules, and they form like a net that helps to prevent blood loss. Okay, so let's go back to this idea of hemostasis. Okay, so hemostasis, so we need to prevent blood loss, as our blood is contained within vessels, and we don't want red blood cells ending up out into the tissues, for example. We want the red blood cells to stay in the vessels and transport the oxygen that way. Okay. So this is hemostasis, would be preventing red blood cells from leaving the circulatory system. Okay. So the stages include a vascular spasm. So vascular spasms. So the blood vessels contain smooth muscles that can constrict and contract. So when the blood vessel constricts, it will shed blood to other parts of the body. So that constriction of the blood vessel is called vasoconstriction. So vasoconstriction is where the diameter of the blood vessel becomes smaller, and this shunts blood elsewhere. So it shunts blood away from the injury. We'll put damaged vessel. Okay. So in your book, there's these stages, and that first one is the vasospasms. So you should be able to, oops, I'm going the wrong way. Okay. So this shows the contraction of the blood vessel, and then the platelets then adhere to the damaged lining of the vessel. So we could then call the platelet plug formation. So you're on the right, number 2, platelet plug formation. And remember, I mentioned that those platelets actually start that enzymatic cascading event, which will ultimately lead to fibrinogen being converted to fibrin. Okay, so the third step is coagulation. So fibrin will form a mesh that traps the red blood cells and the platelets, and then a clot will be formed. Now, the problem that we have with this is we only want blood to clot when it should, right? So if there's a damaged vessel, yes, we want clotting, but we don't want clotting to occur inappropriately, because then those clots can clog up blood vessels like in your brain or in your arteries leading to your heart, and it can lead to things like stroke and heart attacks if we get inappropriate clots or clots being formed. So we'll talk more about that in a minute. Okay, so clotting factors. I am not going to make you memorize all the different clotting factors and what they do, but they're numbered, right? And so they go up to like 12. Is that right? 12? 10. I thought there was 13, but I don't see 13 on there. Anyway, so this is what an enzymatic cascade does. And there can be some diseases like hemophilia. So when we look at hemophilia or hemophilias, this is excessive bleeding. Right? And this can be due to a defective clotting factor. Because all of these clotting factors are produced by and regulated by genes. Hemophilia A is the most famous hemophilia because it was in a royal lineage back when they discovered it. And it is actually due to a defective factor, I think it's 7. Make sure I write that down. 8, sorry. So hemophilia A is a genetic disease, and it is a defect in factor 8. They also have these pathways broken into intrinsic versus extrinsic. So intrinsic has to mean that it's initiated by the platelets. So the platelets play a role in initiating this enzymatic cascade. Extrinsic would mean that there's actually in our tissues, almost all of our cells are capable of responding to damage by producing a tissue factor. And so let's say you got damage in a skeletal muscle. Your skeletal muscle could initiate clotting outside of the platelets initiating it, okay? So that's the difference between the intrinsic and the extrinsic pathway. So we'll watch a little video that talks about this coagulation cascade. Up the volume a little bit.
At the site of vessel injury, the first platelets arrive to start sealing the wound. The coagulation cascade with its various coagulation factors is activated. This involves
Rewind it.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves 2 pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascade meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the 2 pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A, in combination with 5A, activates thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
So that is the idea of an enzymatic cascade. And so at the very end there, we have an enzyme called thrombin. And I just want you to know that that is the enzyme that converts fibrinogen to fibrin. So we'll go back to our notes. And at the end of this, right, this is called thrombin, which is the kind of the ending of the cascade that then ultimately will cause fibrinogen to be converted to fibrin. So write that down again. So okay. So fibrinogen. This is the enzyme, so I'm going to write it up here. Thrombin to fibrin. And I think that this is important because we're going to talk about the difference between an embolism, right, and a thrombus, and a embolism and a thrombus. Okay. Okay, so the last thing I want to talk about is when clotting appears inappropriately. So this is called a thrombus. And this is a clot that forms in an unbroken blood vessel. Okay. This is also adhered to the blood vessel wall. And so it adheres to the blood vessel wall. If it breaks free, it's called an embolism or an embolus. So an embolus is a clot that has broken free. Okay, so this can travel anywhere in the body. And for example, it can could lead to a blocked blood vessel in the brain. And what is that called? Where the blood gets constricted and no longer is able to support the nervous tissue. An aneurysm, and that can lead to a stroke, right? Okay. So this would be bad, right? To have just blood clots in your body. So when you go into the hospital now and you have any kind of medical procedure, what do they do with your legs? They don't elevate them. What are they always doing with your legs when you're not moving? Maybe it's only older people. No, even my daughter, they were doing it. So like after a surgery, you have a surgery, they put you in these really weird compression things. And what it does is it pumps up and puts pressure on your legs, and it feels like, you know, something's pressure on your legs, and then it releases. And so what it's doing is it's kind of mimicking walking, right? Because they don't want blood clots to form in your legs, so they're constantly constricting and relaxing to keep the blood flow moving in the legs. And the same thing goes with like when you're older and some people are more prone to blood clotting, and so like if you're on an airplane, they tell you never to sit for like longer than 45 minutes. You actually have to get up and walk around, so don't feel so bad. You know, those annoying people that are always out of their seats and walking the aisle in the airplane. It could actually be because their doctor is like, you need to get up and walk around, because they could be at risk. If they sit there too long and the blood starts to pool in their legs, they could be at risk of getting a blood clot. So they're just trying to prevent maybe blood clots from forming in their legs. Okay. So one reason that this can occur is that you could have damage to the blood vessel due to infection. For example, infection can lead to damage to blood vessel wall. Okay. So specifically, when you go to the dentist and you have like your gum is swollen up and they freak out on you, right? Like in one little spot, like you'll have this big swollen gum. Why are they freaking out about a swollen gum? Maybe, but what is it that leads to? So what they've done is they've linked the bacterial infections in the gum, which is called periodontitis, so, or periodontal disease, right? So this is swollen gums. Okay, this is related to bacteria that can get into the circulation. And this can lead to heart disease. So specifically, it could eventually possibly lead to a heart attack. So it is related or linked to cardiovascular disease, including heart attacks. If you guys are not old enough to have them freak out on you, but.
So is this why my brother who has heart problems, the dentist won't even touch his teeth unless it's been on an antibiotic for these 2 weeks?
Yeah, so they're like, oh my God, we gotta put you on antibiotics. And then they're like, you have to come right back in. We have to check your gum again. And they're like totally freaking out by that. And that's the reason. Is that if he already has heart problems and his gums are swollen, they want him to be on antibiotics so that if they break a blood vessel and get bacteria into the circulation, it's not going to cause a problem. Okay. Okay, I think I'm going to stop there. So remember that you do have a quiz on Wednesday, and it'll just be over the information since the midterm exam. So just on the information that we've covered in chapter 17.
Are we gonna cover like sepsis?
I don't think it's in your book.